Hello guys, how are you? This is Juan Silva Godinez. Most of you already know me from the PPCR courses. I have been a teaching assistant for the last 3 years, and these are my 2 cents for the TA workshop presentation that we are required to do. As you may realize, I will talk about the coronavirus vaccine. That is, I think, the most spoken topic right now, and I wish to analyze the article that just was published a few days ago. I do hope you enjoy my presentation. Well, as you may know, this is the article that was published on December 31st of 2020. It talks about the efficacy of the Pfizer biotech vaccine, and this is the date it was published. And let us analyze the results to see if they are valid and if we can conclude what we are supposed to do. So this is a screenshot from the results section for the article. I must clarify that I will not discuss the article truthfully, but rather I will focus on the numbers that are important to reach a correct conclusion. I do hope you will follow me since I will be introducing concepts not normally reviewed on the PPCR. So let us take a look at the numbers. So let us take a look at the numbers and rounding for the sake of simplicity. Around 44,000 patients received an intervention, 22,000 in the vaccine group and 22,000 in the placebo group. And with these numbers, there were 8 cases of COVID-19 in the vaccine group and 162 cases in the placebo group. So, with these numbers, the authors concluded that the vaccine was 90% effective in preventing COVID-19 with a credible interval from 90% to 97%. So, what does this mean? What does the efficacy means in this trial? According to these authors, efficacy is the capacity of a given intervention under ideal or controlled conditions. This is the most widely known definition, but the problem with this is that we are not aware of how many patients we have to treat in order to observe a given outcome. The same will go to effectiveness defined by the authors as the ability of an intervention to have a minimal effect on patients in normal clinical conditions. While in PPCR we do distinguish the efficacy and effectiveness concepts, the problem with this is that we are not told how many patients we have to treat to observe the outcome we are looking for. And the same will go to efficiency, which is defined as doing the things in the most economical way. So I think we have to define how can we look at the numbers that we need to know in order to assess how really is the vaccine helping the people. Looking back at the results sections, I have highlighted the magical words in these abstracts. So we are told that the vaccine was 90% effective in preventing COVID-19. What does this mean? The previous statements are the important and tricky part of this article because much to our surprise, the conclusions are misleading. It is not that the vaccine does not work, but rather that we are not given the correct interpretation to analyze how much it works. So let us discuss how to analyze this information. In order to fully understand how effective the vaccine is, we need to introduce this concept, the number needed to treat. The number needed to treat offers a measurement of the impact of any intervention by estimating the number of patients that are needed to be treated in order to have an impact on only one person. Remember that whenever we're running a trial, we are interested in an outcome, whether it be a drug, a surgical, or in this case, a vaccine trial. we define the outcome of interest. And in this particular case, we are interested in the total number of patients that are affected or not with COVID-19 after the vaccination. So in order to assess how effective in reality is the vaccine, we need to calculate the number needed to treat. But before this, we need to calculate another indicator that is known as the absolute risk reduction, which I will define next. The absolute risk reduction is defined as the difference between the exposed event rate and the control event rate. As you may recall, this is a 2 by 2 table that is normally used when we are dealing with categorical variables. Since the outcome of interest is the presence or absence of COVID-19 after the vaccination, this is appropriately used for this case. So let us take a look at the numbers again. So filling in the blanks to build our 2 by 2 table, we take the total number of patients, which will go here. We take the 8 cases in the vaccine group, which will go here. We take the 162 cases in the placebo group, which will go here. And these numbers is the remaining number of patients in each group, which I get subtracting the number of cases from the total of the group. So having done this, we have the numbers required to do our calculations. So now that we have built our 2 by 2 table here, we have the information we require to calculate the placebo event rate, which is equivalent to the control event rate, and our vaccine event rate, which is equivalent to the exposed event rate. So let us see what happens. To calculate the placebo event rate, we take the number of cases in the placebo group and the total number of patients in the placebo group that will go over this like this, and we get this number. This number we will use later to calculate the number needed to treat. And to calculate the vaccine event rate, we take the total number of cases in the vaccine group, which is this, and then again we take the total number of patients in the vaccine group, which as you can see is this. We divide them and we take this number. So we got the numbers we require to calculate the number needed to treat, as the absolute risk reduction is already known. Summarizing our calculations, we know that the number of the placebo event rate is this one, which will go here, and we know the vaccine event rate is this one, which will go here, and the absolute risk reduction, as you may recall, is the difference between these numbers, which is this one. But what does this number means? By itself, the absolute risk reduction may not tell us much information. However, please remember that we calculated the absolute risk reduction in order to be able to calculate the number needed to treat, which is defined as 1 divided by the absolute risk reduction. So in this case, we can finally calculate the number needed to treat, which is this one. And what does this number tell us? As you can see, the number needed to treat is 1,412. This means that in order to prevent just one single COVID-19 case, you need to vaccinate at least 1,412 persons. This number comes in contrast to concluding that 95 out of 100 persons vaccinated will be protected against COVID-19. This is the reason you need to look carefully at the numbers and what they really mean, because as I told you before, the results as they are put in the abstract are misleading. And why does this happen, you may ask? It is because each treatment success depends on the prevalence of the disease you are treating. And this is a very neglected issue when interpreting the results of a trial. The higher the prevalence of a disease in a given place, the more successful our treatment will be. And in contrast, the lower the prevalence, the more people you should need to treat in order to observe just one single positive outcome. And this is always forgotten. In the case of the COVID-19 pandemic, we are always told that the numbers are staggering with a total case burden of around 95 million patients and the total number of deaths of around 2 million, but we never compare those to the total population that is at risk. If we calculate the prevalence considering global cases as 95 millions against 7,800 millions of inhabitants and using this formula which calculates the prevalence as the number of cases over the population at risk, we obtain a prevalence of 12 cases per 1,000 inhabitants anywhere in the world. Just for comparison, and using cancer, which is considered a rare disease, we get a prevalence of 8 cases per 1,000 inhabitants. So remember, for the evaluation of the effectiveness of any treatment, you always have to consider the prevalence of the disease at question. So my conclusions for this presentation will be that we always need to be critical with the numbers we are presented, to remember that the concepts can be tricky. In this case, we were told that the vaccine is 95% effective without being told what does that mean, and that this could lead to misleading conclusions if we do not fully analyze the data we are presented. So remember, always analyze your data. I do hope you enjoy my presentation and see you in the workshop. Bye guys.